Okay, today we are getting into the sliding filament model of contraction. So now we're really going to start looking at how the muscle contracts from the point that it receives a signal from the nervous system to the point that it actually goes through and generates a force within the muscle tissue. So when we look at what the sliding filament model of contraction is, we look at both a relaxed state and a contraction state. In the relaxed state, we're going to see that the thin and the thick filaments overlap only slightly. However, during contraction, we're going to see that the myosin heads are going to bind to the actin molecules and they're going to detach, then bind again. And so they do this repeatedly, as essentially what they're doing is they are pulling and propelling the thin filaments towards that M line, therefore shortening that particular sarcomere. And so therefore then as the H zones shorten and disappear, the sarcomere shortens, the muscle cells shorten, and the whole muscle shortens. This graphic here kind of gives us an overview of the three major steps involved with skeletal muscle contraction. Step one is that we basically have excitation of the skeletal muscle fiber, meaning that we're receiving a signal down the neuron from the nervous system that helps to stimulate the process within the muscle. Our second step then involves the sarcolemma, the T tubules, and the sarcoplasmic reticulum as we get into what we call excitation contraction coupling. And that's going to stimulate the action potential down the muscle cell and down the sarcolemma deep into the muscle cell itself. And then lastly, we get into the myofibrils. And these are going to include the sarcomeres. And it's at this point that we got what we call a cross-bridge cycle. And you'll see that we end up having the myosin heads bind to these actin molecules and pulling them closer and closer together, which causes shortening of the sarcomere, which causes shortening of the myofibril, which causes shortening of the muscle cell, and all the way up the chain. So these are the events of an action potential. Now, when we look at this graph, we are going to make a couple changes with what we have listed here. So with the threshold, our threshold is going to be minus 55 millivolts. So please make sure that we kind of jot that part down here. And then when we look at our resting membrane potential, which we've already touched on and talked about before, we're going to make that 70 millivolts. So again, also make sure that we jot this down as well. So make those corrections to this table and to this graph. So when we look at our threshold, that's going to get us to minus 55. Once we get to this minus 55 portion, if we reach threshold, we're going to generate an action potential. And so as we look at this red line here, this chart, this process where we're actually making the cell more positive is called depolarization. The point where we're making the cell more negative is called repolarization. And then you'll notice there's just a little bit of a dip down here that actually dips below what our resting membrane potential would be. And that's going to be what we call hyperpolarization. And with hyperpolarization, we're typically going to get to a point that's going to be around 90 millivolts. So that's going to be our hyperpolarization phase. But again, it kind of is included in repolarization because repolarization really takes us from the point that depolarization ends all the way until we get back to our resting membrane potential. And so we'll actually talk more about hyperpolarization a little bit later. But these are the two major phases of an action potential in a muscle cell. We have depolarization that makes the cell more positive, and we have repolarization that's making the cell more negative back to its resting phase or resting membrane potential. So what are some of the requirements for skeletal muscle contraction? Well, the first thing we have to have is some sort of activation. We have to have neural stimulation at a neuromuscular joint. We also have to have excitation contraction coupling. And this is the point after the activation where we're actually generating and creating and propagating an action potential along the sarcolemma. Remember, the sarcolemma is the plasma membrane of the muscle cell. And then lastly, we have our final trigger. And that final trigger is going to be a brief rise in intracellular calcium levels within the muscle cell itself. So these really are these three phases that we just kind of touched on in the earlier graphic. Our three phases: the activation at the neuromuscular junction, excitation, contraction, coupling, and then the final trigger is a brief rise in calcium, which leads to our cross-bridge cycles. So the events that occur at the neuromuscular junction. So as we go through these, we're going to go through them in the slides here. And then after that, we'll actually pull up a graphic so we can see what really happens. I will have some videos for you that should be listed in the videos page. And I will also try to add them into the module for this particular section. So when we look at the skeletal muscle is going to be stimulated by somatic motor neurons. Remember, when we talk about somatic, we should think skin, skeletal muscle, and joints. However, when we look at this term here, this term motor basically means movement or some sort of active control. And so when we look at joints don't move by themselves, they require muscle. So joints basically just have sensation or sensory input. When we look at the skin also really doesn't have contraction. It simply is a means or a medium. And so it also really only has sensory input. And so when we look at the motor component of the somatic classification, somatic motor neurons are neurons that go to innervate skeletal muscle and cause movement. The axons of the motor neurons, which are the part that go away from the cell body of the neurons, the axons of these neurons are going to travel from the central nervous system via nerves to our skeletal muscles. And then each axon is going to have several branches as it splits off and enters into the muscle. Each of these axon endings or axon terminals are going to form a neuromuscular junction with a single muscle fiber. So every single axon splits off, and every single muscle fiber, every single muscle cell has a neuromuscular junction with an axon terminal. So then what happens is the nerve impulses are going to arrive at the axon terminal. The next step involves our first type of voltage sensitive channel. And so we have voltage gated calcium channels. And what these voltage gated calcium channels do is they open up. And this allows calcium to enter the axon terminal. This causes acetylcholine to be released by exocytosis from our synaptic vesicles. And these synaptic vesicles are the structures that are actually going to contain the acetylcholine. This then allows acetylcholine to cross the synaptic cleft as it's released by exocytosis from the synaptic terminal, and it's going to bind with the receptors on the motor end plate. These electrical events that occur then are actually going to lead to the generation of an action potential. And we'll talk about those steps here coming up. So if we take a look at this first phase, you'll see that here we have our motor neuron, and here we have our synaptic knob and axon terminal. You'll notice that within that axon terminal, we have a bunch of synaptic vesicles. Within it is contained acetylcholine. And so what happens is we have our action potential, which is our impulse or our signal that comes from the brain down the axon, and it arrives at the axon terminal. An action potential is basically a change in voltage. And so we have these, we have this change in voltage here that now stimulates the opening of our voltage gated calcium channels. And so as our voltage gated calcium channels open, we have this flood of calcium into the axon terminal. And so I'm actually just going to draw an arrow here. So we have calcium flooding inwards here. So calcium floods in, and it causes our synaptic vesicles now to move towards the edge of our synaptic terminal. Now, as these vesicles reach the edge of our axon, they're actually going to release their contents, which is acetylcholine. And acetylcholine crosses this synaptic cleft here and is now going to bind to the channels that we find on the motor end plate. Now, these channels here are going to be our ligand-gated sodium-potassium channels. And these ligand-gated sodium-potassium channels are going to open in response to acetylcholine binding to them. Now, when those open, and I'm getting a little bit ahead of myself, but we'll touch on this here as we go through this. But one thing that we're going to see then is sodium is going to flood in, potassium is going to flood out. So we have sodium in, and then at the same time, we have potassium out. Now, this occurs, as far as sodium goes, occurs at a greater rate going in than what we see with potassium moving out. And because we have more potassium, or excuse me, we have more sodium moving in, we're actually going to see a change in voltage on this motor end plate. And because we have more positive ions coming into the cell than what are leaving, we end up seeing that the change in voltage becomes more positive. So in order for the process then to stop, we have to basically break down or get rid of acetylcholine. Now, acetylcholine is often broken down due to an enzyme, and that enzyme is called acetylcholine esterase. And what acetylcholine esterase does is it breaks down acetylcholine into its component parts of acetic acid and choline. And what it does then is it prevents continued muscle fiber contraction in the absence of additional stimulation. So essentially, when that stimulation of the action potential stops, it removes acetylcholine so that our ligand-gated channels are able to close up, and that is a way that we're able to stop the contraction on the muscle cell. So when we look at a clinical view of this process, or that affects this process, we look at what we call myasthenia gravis. Now, myasthenia gravis is an autoimmune disease that we primarily see in women. And so the receptors are essentially removed from the muscle fiber by endocytosis. And so as antibodies basically break down or remove the receptors, that means fewer receptors at that location for acetylcholine to bind to. And this is going to result in a decreased stimulation of the muscle cell. It causes rapid fatigue and muscle weakness. And oftentimes we see the eye and facial muscles involved first. And it can also then be followed by swallowing problems and limb weakness. So now let's get into the second phase of muscle contraction here. So we basically did the stimulation portion, the action potential down the neuron. Well, now we get into the excitation contraction coupling phase that occurs within the muscle cell. So we have the stimulation of the muscle fiber is actually coupled together with the sliding of the muscular filaments. The coupling includes the end plate potential, the muscle action potential, and the release of calcium from the sarcoplasmic reticulum. So let's look at the first few steps in this process. So one of the things that we have to do is be able to generate an action potential after it arrives from the nerve cell, because now we have this space, this synaptic cleft, where we're actually crossing with our acetylcholine. And so in order to have a connection of some sort, we have an indirect passage of that action potential from nerve cell to muscular cell. And so in order to get to that point, we go through a local depolarization. Now, the point to a local depolarization means it doesn't necessarily get us an action potential. One thing we're going to talk about when we talk about action potentials is that an AP is an all or none event. It either happens or it doesn't. But in order for it to happen, it must reach the threshold stimulus level of that minus 55 millivolts. We can have a local depolarization event which doesn't actually reach threshold. Let's say it goes from minus 70, which is our resting membrane potential, and it goes to minus 60. That's a depolarization because it's making the cell more positive, but it's not enough to actually cause an action potential. And so when we get these local depolarization events, we call this an end plate potential. Okay? And so what ends up happening in order to get this local depolarization or to create this end plate potential is our acetylcholine binds to our ligand-gated sodium-potassium channels. Those channels now open up, and we have this simultaneous diffusion of sodium inward and potassium outward. So we have simultaneous diffusion of sodium inward and potassium outward. Make note that, again, we said this before, there's more sodium that diffuses in. So the interior of that cell and the sarcolemma actually becomes less negative, or what we would call more positive. And this then is a depolarization. And the depolarization is initially a local event called an end plate potential. Okay? So again, locally at this particular location, it's called an end plate potential. The second phase then of this is to generate and propagate an action potential. So the end plate potential now begins to spread as more and more channels are open, more and more sodium comes in across the entire end plate. And so it's going to spread to these areas adjacent to it. As it leaves the motor end plate and actually gets out onto the rest of the sarcolemma, it's now going to stimulate our third type of channel. And that third type of channel is our voltage-gated sodium channels, okay? And so voltage-gated sodium channels are going to open in response to a more positive change in the plasma membrane charge. And as it becomes more positive, these channels open up and we have an increased flood of sodium into the cell. We can guess, based on what occurred at the motor end plate, that an increase in sodium into the cell continues to make more areas of that cell more positive. And so the sodium influx decreases the membrane voltage toward a critical threshold, and therefore, if the threshold is reached, an action potential is generated. So then what happens is this local depolarization wave continues to spread, and it changes the permeability of the sarcolemma by, again, allowing more and more sodium to flood into the cell. The voltage-regulated sodium channels open in adjacent patches, causing the next area of the cell to depolarize to threshold. And this continues all the way down the entire sarcolemma. So as we have this wave of depolarization moving down the sarcolemma of the muscle cell, literally right behind it, we have what we call the repolarization wave. And so as our sodium channels begin to close, we now start to see voltage-gated potassium channels begin to open. And so as voltage-gated potassium channels open, we have this massive efflux of potassium. And as we release these positively charged potassium ions, we have positive charges leaving the muscle cell. This is actually going to make the cell more negative. And so this rapidly restores the resting polarity, getting it back to its resting membrane potential of minus 70. In this particular part of the phase, the fiber cannot be stimulated and is in what we call a refractory period until repolarization is complete. We also are trying to restore the ionic conditions of the resting state, and this is accomplished by utilizing the effects of the sodium-potassium pump. So if we take a look at this, you get a chance to see the entire graphic here from essentially from start to finish. So we see phase one, it's not indicated here, but it's the action potential that comes down our neuron. As the action potential comes down, it stimulates our voltage-gated calcium channels. From here, calcium floods into the nerve cell, particularly into this axon terminal. The flood of calcium forces the synaptic vesicles to move towards the end of the axon terminal, and it forces the vesicles to release their contents by exocytosis into the synaptic cleft. The contents, which include acetylcholine, cross the synaptic cleft then and bind to the receptors on the motor end plate. Okay, so I think we have a bigger graphic here. So here we have our acetylcholine binding to our ligand-gated sodium-potassium channels. Again, here is identifying our motor end plate, and so sodium floods in, making this particular section more positive. This is the depolarization phase, the local event that we call the end plate potential. If we have an increase of enough sodium to increase our resting membrane potential up to the threshold stimulus, we now end up getting what we call the propagation of that action potential. And so that change in voltage up to threshold of minus 55 now actually causes our voltage-gated sodium channels to open, because again, they open in response to a change in voltage towards the positive side. So now we have this massive influx of sodium outside of our motor end plate. And you'll notice that we have these voltage-gated sodium channels throughout the sarcolemma here. So we have a continued increase in sodium coming into the cell, which now makes the cell more positive, and we continue that wave moving all the way down the sarcolemma. Now, as we reach the peak of this action potential, we then start to stimulate our voltage-gated potassium channels to open up. As these channels open, we now start to see a decrease in our voltage. And so now we're making the cell more negative. And this returns us back to our resting, or aids in the return back to our resting membrane potential. And when these channels open, this is what we know as repolarization. So now as that action potential continues to move down the sarcolemma in this direction, you'll notice that we're actually heading into the T-tubule. Now, I want you to understand this arrow. This arrow is not necessarily outside of the cell. This arrow represents the transmission of these charges, of these ions, down the sarcolemma here. And so as it moves this direction, it moves down the sarcolemma. Remember, the T-tubules are continuous with the sarcolemma. So now we're transitioning into or down the T-tubules. We have these voltage-sensitive calcium proteins that get stimulated by the change in charge, by the change in voltage. And so as they are stimulated, they cause these voltage-gated calcium channels that are located on the sarcoplasmic reticulum to open up. And this allows a flood of calcium into the cell of the muscle. And this leads us then into our cross-bridge cycle next. If we take a look, or we kind of box this all together then, here we have a terminal cisternae of the sarcoplasmic reticulum. Here we have a terminal cisternae of our sarcoplasmic reticulum. And here we have our T-tubule. Again, these three structures together make up a triad. So again, here are the events of an action potential at the sarcolemma. We have our resting membrane potential, again, is minus 70. Our threshold is minus 55. We have depolarization, in this case at the end plate, and so we call it an end plate potential. If it reaches our threshold, we basically call this then an action potential. And again, an action potential is an all or none event. So we're moving it from minus 55 and it's going to spike all the way up to positive 30. It's at this point then we begin repolarization and we start to change the charge. In this phase here, in phases 2 and 3, phase 2, we basically are at our end plate. And so these are our ligand-gated sodium-potassium channels that are opening here. We then stimulate the voltage-gated sodium channels kind of in this number 3 here. And so this occurs as the voltage-gated sodium channels on the sarcolemma open and sodium enters rapidly. And then in phase 4 here, you see in repolarization, we now have our voltage-gated potassium channels that begin to open up. And this causes a change in voltage becoming more negative back towards our resting membrane potential. This table or this graph is a really good study tool to really start to understand what channels are opening during the process here. Now, as we start to transition down the sarcolemma, we now start to utilize or look at the sarcolemma, the T-tubules, and the sarcoplasmic reticulum. This is kind of that next phase of the excitation-contraction coupling process. And so our action potential travels down our T-tubules. And as it travels down the T-tubules, it causes our voltage-sensitive calcium channels in the T-tubule membrane to trigger the opening of our calcium release channels in the sarcoplasmic reticulum terminal cisternae. And the release of calcium from the terminal cisternae actually goes into the cell, and this interacts, or the calcium interacts with our myofilaments, and this is the process that actually triggers the contraction process of the muscles. So this then leads us into one of our last phases of this process, and that's called the cross-bridge cycle. So as calcium binds to troponin then, troponin is a protein that now goes through a shape change. And as it changes shape, it actually causes the tropomyosin to shift off of our myosin binding sites of the actin molecule. So when we look at cross-bridge cycling then, we really have 4 repeating steps that occur over and over and over in a cycle until the signal stops. Those four steps are called cross-bridge formation, the power stroke, the release of the myosin head, and the reset or the cocking of the myosin head. So when we look at these steps, the first one is the cross-bridge formation. In the cross-bridge formation, the myosin head attaches to the exposed binding sites on the actin molecule. After we form that cross-bridge, we now pull the thin filament. And so the myosin heads basically pull the thin filament toward the center of the sarcomere. And this decreases the distance of the H zone. At this point then, ADP and inorganic phosphate are released. And in order to release the myosin head from the actin molecule, we have to have ATP come in. And ATP binds to the myosin heads. And it's this binding that actually causes the release of the myosin head from the actin molecule. We then split the ATP back into ADP and inorganic phosphate. This helps to reset or cock the myosin head, and it provides us the energy to put that myosin head back into the reset position. And now it's ready to start the forming of the cross bridge all over again. And with that, we're going to use cross bridge as our attendance code word for today. So again, the attendance code word for today's lecture is cross-bridge. So our cross-bridge cycling then will continue as long as calcium and ATP are present. As this process continues, it results in the sarcomere shortening as the Z-discs move closer together, and then it begins to narrow or cause a disappearance of our H zone as well as our I band. Now, it doesn't change the length of the thick and the thin filaments, because those lengths or those filaments themselves remain the same length. However, they do start to slide past each other. And that's the reasoning for the disappearance of the H zone and the shortening of our sarcomere. The next slide that we go over is going to show in graphical representation of our cross-bridge cycle. So hopefully it kind of puts all these steps into perspective. So you can see we take this window up here in this upper left corner of your screen, and we kind of move it into a larger window so we can see it a little bit better. So here you'll notice we have our calcium ions. In blue here, we have our troponin, and in green, we have our tropomyosin. And here in red are the actin subunits. And then you'll see the black little dots. Those are the myosin binding sites on the actin molecules. So as you see calcium comes in, it's going to bind to troponin. Now troponin is going to go through a shape change. And as it changes shape, it causes our tropomyosin to shift off of these myosin binding sites of the actin molecules. And this now allows a location for the myosin heads to come in and bind. And so if we look at the process here, we now then lead to, once our myosin binding sites are open, this now leads us into the cross-bridge formation. And so we go through this formation by going through a process that hydrolyzes ATP. And so as ATP comes in, we go through the process, we break it down into ADP, an inorganic phosphate. We use that energy then to go through what we call the power stroke. And so our myosin heads basically pull the actin molecule towards the M line, and it decreases the distance of the H zone. So the myosin head then swivels towards the center of the sarcomere, pulls the attached thin filament, and this motion again is called the power stroke. And it's also at this point, the ADP and inorganic phosphate are released. In order for the myosin to actually release itself from the actin, we need an ATP molecule. So as the ATP comes in and binds to the myosin head, that causes the release of the myosin head from the actin molecule. And then we restart or reset that myosin head. And so now the ATP then is again split. We now have ADP and inorganic phosphate, and now we're basically ready to go through the power stroke all over again. And so by utilizing or creating energy from ATP, we now are able to come back, bind to the actin molecule, go through the power stroke and release ADP, release the myosin head as ATP comes in and binds, and then we come back and we cock the myosin head all over again as ATP is broken down. And that process just cycles through and continues until the contraction is over. And once that contraction is over, then the myosin head obviously releases. It is back to its cocked position and ready to bind to it again. The tropomyosin comes back. It covers the myosin binding sites on the actin molecule. Troponin resets back to its original conformation, and calcium is released from troponin. And so this is the process of our cross-bridge cycle. And again, it is a continuous process until the contraction signal is stopped. When we look at the sarcomere and we start to shorten it, this is actually what we start to see. Again, you see this overlap in a relaxed state of the actin overlapping the myosin. But as we go through contraction then, you'll notice here's our Z disc and here's the other Z disc. So this is the length of our sarcomere. You'll notice that in this case, in a contracted state, it's shortening. You'll notice that as we pull our thin filaments together, we now have a disappearance of that H zone. And this also then leads to the shortening of our sarcomere. So as we look at a clinical view, we look at muscular paralysis and neurotoxins and how these affect muscular contraction. Two particular neurotoxins that we talk about are tetanus and botulism. Now, tetanus causes spastic paralysis. And so spastic paralysis is this kind of relatively violent contraction of the muscle cells themselves. It causes spastic paralysis by the toxin Clostridium tetani, and it blocks the release of the inhibitory neurotransmitter in the spinal cord. And this leads to an overstimulation of the muscles, which basically gives us that kind of almost like Charlie horse type of effect. But again, we don't really have a lot of control over it because, again, it is a type of paralysis. Our second one is botulism. Now, botulism causes muscular paralysis, but it's caused from the toxin Clostridium botulinum. And what it does is it prevents the release of acetylcholine at the synaptic knobs. And so you can imagine that instead of having this spastic paralysis, we have more of a relaxed type of paralysis. And since we're preventing the release of acetylcholine, we don't have the ability to open up our ligand-gated sodium-potassium channels. Now, botulism can be life-threatening if it's ingested, and especially if you get enough of it. However, it's also used to treat certain conditions. We use it to treat injections for cerebral palsy, or it can actually be used in cosmetic purposes for diminishing wrinkles. So this is what we know as Botox. We actually inject the botulism toxin into our tissue, and that causes a relaxation, in this case, of the facial muscles. So now that we went through muscular contraction, now we have a chance to really go back the opposite way. And so we look at the events in muscle relaxation. So now the events in muscle relaxation are going to include the termination of the nerve signal and acetylcholine release from the motor neuron. So that process stops. We also have the process of hydrolysis of acetylcholine by acetylcholine esterase. And this basically breaks down acetylcholine into its component parts of acetic acid and choline. Because we no longer have acetylcholine in that particular part of the synaptic cleft, we have the release, or we have the closure of our acetylcholine receptors, and this causes a stoppage of the end plate potential, which means we have no further action potential being generated down the sarcolemma, which then allows us to close the channels in the sarcoplasmic reticulum. This allows our calcium to be returned back into the terminal cisternae. It causes the troponin molecule to return to its original shape. It causes the tropomyosin back to the blocking of the myosin binding sites, and it allows the muscle then to return to its original position due to the elasticity of the muscle.